Myself Manjunath M. Second year m.sc, Government Science College, Asan. My seminar topic is Significance and Measurement of DO, BOD and COD. Submitted to Department of PG Studies in Chemistry, Government Science College, Asan. Under the supervision of Dr. Pramila SC, Assistant Professor, Department of PG Studies in Chemistry, Government Science College, Asan. First, we discuss dissolved one by one. Dissolved oxygen. Dissolved oxygen, the greater the amount of oxygen dissolved in water, the better is the quality of water and thus it will give better support to the aquatic life. The dissolved oxygen in the range of 4 to 6 ppm will support plant and animal population in any water body. Water with less than 2 ppm DO will support mainly worms, bacteria and fungi. Significance: just as we need air to breathe, aquatic organisms need DO to respire. It is necessary for the survival of fish, invertebrates, bacteria and underwater plants. DO is also needed for decomposition of organic matter. Measurement of dissolved oxygen. Winkler method. Reagents required in the measurement of dissolved oxygens are standard sodium thiosulfate, water sample, manganese sulfate, alkaline Ki, sulfuric acid and starch indicator. Procedure: standardization of sodium thiosulfate. 10 ml of 0.02 normal potassium dichromate solution was pipetted out into a conical flask. Add 10 ml of sulfuric acid and 5 ml of potassium iodide solution was added. The liberated iodine was titrated against sodium thiosulfate solution using starch indicator till the endpoint changes from blue to colorless. Volume of sodium thiosulfate is noted and strength of sodium thiosulfate is obtained. Titration with water sample. Transfer the water sample from 50 ml capacity glass topped bottle to 250 ml conical flask and 2 ml of manganese sulfate solution by pipetted out just below the surface of water added and 10 ml of alkaline Ki was added and the stoppered conical flask was shaken well and keep aside for few minutes and allowed to precipitate to settle and 1.2 ml of concentrated sulfuric acid was added and shaken thoroughly for few minutes and titrated against standard Hypo solution. The sodium thiosulfate is also called Hypo solution. When the color of the solution is slightly yellow, 2 ml of freshly prepared starch was added. Turns to blue. Continue the titration until the blue color changes to colorless. Repeat the titration process to get the concordant value. Calculation: normality of Hypo solution, which is equal to normality of sodium thiosulfate into volume of potassium dichromate whole divided by volume of sodium thiosulfate consumed. To give the normality of sodium thiosulfate, mass of oxygen dissolved equals volume of sodium thiosulfate into normality of sodium thiosulfate into equivalent weight of oxygen into 1000 whole divided by volume of water taken in ml to give the mass of oxygen dissolved in milligram. Next, we discuss biochemical oxygen demand. Biochemical oxygen demand is the amount of oxygen required by microorganisms to carry out the oxidation process of organic matter. BOD and DO are indirectly related to each other. When the BOD is too high, the DO becomes too low to support the living organisms. Significance: BOD is commonly used to carry out the water quality measurements. When there is enough bacterial activity, there is the depletion of oxygen in water downstream. This is called oxygen sag. Biochemical oxygen demand represents quantity of oxygen required by bacteria and other microorganisms during the biochemical degradation and transformation of organic matter present in wastewater under aerobic conditions. Reagents used in the BOD measurements are water sample, magnesium sulfate, calcium chloride and ferric chloride. Procedure: preparation of dilution water. Take desired volume of distilled water in a suitable bottle and add 1 ml of phosphate buffer, 1 ml of magnesium sulfate, 1 ml of calcium chloride and 1 ml of ferric chloride. Dilution technique. The dilution depends on the nature of the sample. 0.1 to 1% for industrial wastes, 1 to 5% for raw and settled sewage, 25 to 100% for polluted river water, and 5 to 25% for oxidized effluents. Procedure: aerate the water sample thoroughly by bubbling air through a diffusion tube into this sample for 5 minutes or until the DO is 7 ppm. Make a measured dilution with water if BOD is greater than DO level. Seed with a little diluted domestic wastewater. Measure DO on a suitable aliquot, which is D1. Fill the screw-topped incubation bottle to the brim with the remaining diluted sample. Seal the bottle and incubate in the dark for 5 days at 20 degrees Celsius. Measure DO on an aliquot of the sample, which is D2. Calculation: POD equals D1 minus D2 whole minus B1 minus B2 into F whole divided by P, where D1 is the DO of diluted sample, D2 is the DO of diluted sample after incubation, where B1 and B2 are DO of dilution water containing seed before and after incubation, where F is the ratio of seed in sample to seed in control, where P is the decimal fraction of sample used. Next, we discuss chemical oxygen demand. Chemical oxygen demand is the amount of oxygen required to carry out the oxidation process of organic matter chemically. It is a rapid test for determination of the total oxygen demand by organic material present in the sample. Since many organic materials are chemically oxidizable but not by biochemical means, then COD values do not indicate whether the organic matters present in sample is biodegradable and at what rate biological oxidation would proceed. Significance: COD is required for oxidation process in certain inorganic compounds like sulfides, thiosulfates, ferrous ions and other metals in the reduced state. Reagents used in the COD measurements are standard sodium thiosulfate, potassium dichromate, potassium iodide and sulfuric acid. Procedure: first, standardization of sodium thiosulfate. 10 ml of 0.1 normal potassium iodide solution pipetted out into a clean conical flask. 10 ml of 2 normal sulfuric acid and 5 ml of potassium iodide solution was added. The liberated iodine was titrated with Hypo solution, where Hypo solution is sodium thiosulfate, using starch indicator till the end point is colorless. Volume of sodium thiosulfate was noted and strength of sodium thiosulfate was calculated. Estimation of chemical oxygen. Transferred 50 ml of water sample in 100 ml of conical flask. Carried distilled water as a blank. Then 5 ml of potassium dichromate solution was added to each flask. Place the flask in water bath at 100 degrees Celsius for 1 hour. Cool the flask for 10 minutes. Then 5 ml of potassium iodide and 10 ml of sulfuric acid was added to each flask. Titrated the content against 0.1 normal sodium thiosulfate until pale yellow color disappears. 1 ml of starch indicator was added, then titrated against sodium thiosulfate until blue color disappears completely. Read out the total volume of titrant consumed and took the mean of 3 flasks of each flask. Read out the total volume of titration used for blank titration. Calculation: COD, which is equal 8 into normality of sodium thiosulfate minus S minus B, where S is sample titration value and B is blank titration value, to give the COD in milligram per liter.